Okay, our next video is pyruvate dehydrogenase. We have now looked at glycolysis, going from glucose to pyruvate. We've looked at gluconeogenesis, going from pyruvate back to glucose. And now this video is going to talk about a different fate of that pyruvate. What else can happen? And on our picture here, here's our glucose, here's our pyruvate. And in this case, we're going to be looking at how pyruvate can be converted to acetyl-CoA. After we form that acetyl-CoA, it'll enter the citric acid cycle to become fully oxidized. But for this video, we are just looking at this step, this pyruvate dehydrogenase. That's our abbreviation. Just going from pyruvate to the acetyl-CoA. So as the pyruvate is going to enter this enzyme complex, this pyruvate dehydrogenase complex, and in that active site, 2 electrons are going to get carried away by the NADH. We'll take a look at that. And a carboxylate is going to get kicked out. If we look, here was our glycolysis, here was our gluconeogenesis, and now here is our reaction to get to acetyl-CoA, and you'll see it's a one-way arrow. This is irreversible in animal cells. Once it gets to acetyl-CoA, it can either enter the citric acid cycle, or it can be used for this formation of fatty acids. But for now, this piece right here is what we're going to look at. And since it's one way, once pyruvate enters this enzyme complex, it is committed to forming acetyl-CoA. There's no going back. So let's take a look at this. Here we have our reaction. So pyruvate is going to react with this coenzyme A and NAD+. We're going to get our product, acetyl-CoA, plus we'll produce some waste carbon dioxide. We'll have this NADH and H+. If you remember, our pyruvate is an alpha keto acid. So here's that ketone part, right? Here's our carboxylic acid piece. The ketone is bound to that alpha carbon right there. And this piece right here of our pyruvate is going to get added on to our coenzyme A. So the part in black here is all from the coenzyme A. And this part in pink for acetyl-CoA came from right here. Here, we can circle that in pink. Right here is going to get added on there. And we're going to take a look at how this happens. Okay, we have this thioester linkage. Okay, it is thermodynamically unstable. It's a really high energy bond, but it's kinetically stable. It's got a high activation energy. It requires an enzyme to get past that. Okay, if you kind of remember from this coenzyme A, over here we had that adenine nucleotide ADP plus this extra phosphate group on it right down here. We had vitamin B5 in here, and then we've got this beta-mercaptoethanol amine. But key thing here is just that, remember, we've got these coenzymes, whoops, that is not what I wanted. So B5. So we have like this piece from our diet, right? That we need to be able to have all the pieces for these reactions to happen. All right, so this reaction is going to happen in the mitochondria. when we form pyruvate, it is actually forming out in the cytosol. So when we go through glycolysis and we form pyruvate, it's out in the cytosol, and so it will need to get into the mitochondria. Okay, remember pyruvate's this little charged molecule. It's going to need a carrier. That carrier is called mitochondrial pyruvate carrier. And it's a transmembrane protein that carries the pyruvate into the mitochondria. Okay, once inside, this is where it comes into contact with the pyruvate dehydrogenase complex. So our PDH complex is in the mitochondria. Okay, and it'll be converted into acetyl-CoA. So this reaction will happen. We'll make our acetyl-CoA. And then the citric acid cycle also occurs in the mitochondria. So we're kind of set there. If you think about the mitochondria, it has this double membrane. The outer membrane is pretty porous. Okay, things can move through it largely by diffusion. It's the inner membrane where we have this mitochondrial pyruvate carrier, this carrier to move the pyruvate into the mitochondria. Okay, so synthesizing acetyl-CoA from pyruvate requires 3 enzymes and 5 coenzymes, and it's got 3 chemical steps. Dehydrogenase, when I say dehydrogenase, you should think, ooh, redox reaction, so we know we're going to have redox going on. If we look at our 3 steps, first we're going to have a decarboxylation, all right, so we're removing that CO2. We're going to oxidize that, remove some electrons, and then we're going to transfer that group onto that coenzyme A. So these first 2 steps happen in one of the enzymes. This third step happens in another enzyme. And then that third enzyme regenerates the various cofactors. And we're going to take a look at that in more detail. All right, if we look at the pyruvate dehydrogenase complex, and this says E. coli, but it is going to be largely similar in mammalian cells. And this is a huge complex, millions of Daltons heavy. It is very large. It has 30 subunits of E1, so not just one subunit, but 30. It's got 60 subunits of E2. It's got about 12 subunits of E3. Depends on the specific organism, but these numbers are generally correct for mammalian cells. Okay, these things are huge. The second enzyme right here does exist as a dimer, and so really our ratio is still kind of 1-to-1. We get 1 E1, one of these for every one of these dimers, and we'll take a look, okay? But before we do that, these things have prosthetic groups associated with them. Okay, a cofactor that's tightly associated with the enzyme. They also have co-substrates, acetyl-CoA, NAD+. But I want to take a look at a couple of these prosthetic groups because it turns out that they will end up being important. Okay, TPP, thiamine pyrophosphate. So when we look at this, it has thiamine, which is essentially vitamin B1. Okay, so another vitamin with the pyrophosphate, these 2 phosphates bound together. And this compound will temporarily bond carbon subunits as they go through the synthetic mechanism. The active spot is right here. Really, that's the active piece that's going to bind those carbons. And we'll take a look at how that works. So this TPP molecule is part of the active site and will temporarily bind to the acyl group to form this covalent intermediate. All right, another prosthetic group we have over here is this lipoic acid. And when we look at this, this is also a vitamin, and it's going to bind to a lysine subchain, and it'll be attached to the second enzyme by an amide linkage. A lot of words. Let's take a look at it. Okay, so this lipoamide is formed by the attachment of the lipoic acid, right? So this is what we just looked at right up here. Okay, turned on its side. And this piece right here is going to react with our lysine side chain, and we're going to get right here, we're going to get that amide linkage. Okay, the active part of this, once we get down into looking at our reaction, is this disulfide bond. Okay, up here at the top, here's kind of our polypeptide chain. It would continue out that way, and then this was that side chain that we bound to. And we get essentially a different color. This thing, we're going to call it a swinging arm. Okay, and we looked at that one time before. It's this flexible linkage that allows the lipoamide to move between different active sites. So this disulfide bond down here is going to pick up an acetyl group and carry it from enzyme 1 to the active site of enzyme 2, where it'll undergo reaction to form acetyl-CoA. All right, let's look at that with this kind of cartoon drawing. So our pyruvate dehydrogenase complex in yellow, we have enzyme 1. In pink, we have enzyme 2. And remember, we said they existed as a dimer. In green, we have enzyme 3. And there won't be just one of these subunits. There'll be numerous of these subunits in each of these complexes. So lots and lots and lots of these sets. Okay? So if we think about our enzyme, here we have our enzyme, and pyruvate enters the enzyme and bonds to that TPP. Okay, we form a covalent bond. If we scroll back up here, it's going to form right here. It's going to bond to that. And when it does, it loses that CO2. Okay, the carboxylate group leaves, and that energy from it leaving allows it to make this new bond right here. And so now we've got that group bound to our TPP. So then this swinging arm down here is going to come up here. And interestingly, there's not just one swinging arm per complex, but there's multiple, maybe about 80 per complex. And so this can happen over and over and over. But this swinging arm is going to come up here. All right, and when it does that, we're going to see that sulfur is going to form a bond with the carbon. So we're going to get a bond here. Okay, the sulfur is going to be reduced, the carbon will be oxidized back to a carbonyl, and here we are down here. It gets pulled back down into the enzyme 2 section. Okay, so that swinging arm swings it down into the active site of enzyme 2. The coenzyme A is going to enter, and as that coenzyme A is going to enter, it's going to pick up this acetyl group, okay, and leave as acetyl-CoA. So it's leaving as acetyl-CoA. As that happens, we now have almost, but not quite, our original swinging arm back. Okay, but we need to reoxidize those sulfurs so that it's ready to go again. So these will be oxidized by the FAD. So the FAD will pick up those electrons. We get FADH2. We have our original swinging arm back. And then NAD plus is going to come in and pick up those electrons to get us back to our starting point. Okay, so that NAD plus will take those electrons and can carry them over to our electron transport chain. All right, that was a lot. Step by step, you can sit down and go through these later. Let's talk a little bit about how it is regulated. And it is regulated in two different ways. The first way is through covalent modification. So enzyme 1, so remember this yellow one, okay, is deactivated when it's phosphorylated and activated when not phosphorylated. So there is an enzyme or a kinase, okay, specifically associated with the E2 and E3 parts of the complex. And it will phosphorylate enzyme 1, okay, to deactivate it. So think about when we would want this deactivated. So we know in our glycolysis, we were going from glucose to pyruvate, and we can go back this way, or now we're looking and we can say we can also take in this irreversible reaction pyruvate to acetyl-CoA. So we would not want to use our pyruvate to form acetyl-CoA if we have a need for glucose. So when glucose is low, just like in our last step when we talked about gluconeogenesis, your body will make glucagon, okay? It wants to save the pyruvate to make glucose. So that glucagon will also activate this kinase to phosphorylate the enzyme, the first enzyme, and deactivate it. So when blood glucose is low, we would not want to use up our pyruvate this way, okay? We also have a phosphatase associated with this, okay? And so it will remove that phosphate to allow us to continue this step, to reactivate this. Okay, this can be activated by different things depending on the cell type. So for example, insulin. So when you're in the fed state, you've got high levels of insulin. We could remove that phosphate because we've probably got plenty of glucose, and we can send our pyruvate down to the acetyl-CoA. Okay, another one is calcium. So calcium is generated in muscle cells, released upon contraction in the muscle cells. And so if you think about that, if you are using your muscles a lot, generating a lot of calcium, you're going to need a lot of energy. And so we're going to want to take the pyruvate down to that acetyl-CoA to feed into that citric acid cycle, which we will see will help us with our energy needs. Okay, so one way this is regulated is covalent modification. The other way is allosteric regulation based on the energy charge. Okay, so if we've got plenty of energy, we do not need to make more energy. So we have high levels of ATP. We've already got a bunch of acetyl-CoA or NADH. Those are going to inhibit the PDH complex. Okay, we don't need to keep doing that. We should instead maybe build up our glucose stores, our glycogen stores. On the other hand, if we have low energy, okay, then those will stimulate the complex, and we'll see it move forward. Okay, let's talk about this real quick. We want to think about some advantages of organizing the enzymes that catalyze the formation of acetyl-CoA from pyruvate into a single large complex. All right, so you might pause for a minute and think about what you think those advantages would be. Think about why that would be helpful to your body. This is an important piece. So we have this complex held together by these strong IMFs, and they have to be held together for them to be functional. And this idea, we see it across all living things, these enzyme complexes. So clearly they're important. If we think about why, One would be that the reaction would be facilitated, right? So reaction facilitated by the close proximity. We don't have to wait for one intermediate or something to diffuse out and float over to the next enzyme. They're all right there close together. And then on top of that, that lipoamide swinging arm, it also speeds up the process. So rather than waiting for something to diffuse from one active site to the other, this would speed up the process. Okay, reactants don't have to leave the complex. Reactants don't leave complex. So they're kind of isolated in here if you think about it. They're shuttled from reactive site to reactive site, so this is going to reduce side reactions that might have occurred if it was out in the general area, so it's more efficient. Okay, the enzymes are in the correct ratio before it even starts. You don't have to wait for, maybe you made plenty of E1 and E2, but somehow E3 was slow to make. So we already have everything right there in the correct ratio. Okay, and regulation is efficient because those regulatory enzymes are part of the complex. Regulatory are part of the complex. All right, that is it. Next up, we are going to talk about the citric acid cycle.